Alright, so the diversity of immunoglobulins and MHC antigen presenting molecules is controlled by the human genome. So to start, the typical gene is consisted of exons, which are sequences of base pairs that are transcribed into mRNA and are separated by introns, which are sequences of base pairs between them that are removed in transcription. All cells in the body contain the same genes except for like lymphocytes, which contain genes that undergo a series of recombinations to provide them with a large diversity. So to start with the creation and diversity of immunoglobulins, first I'll talk about the general features of an immunoglobulin. So an immunoglobulin genome is composed of genes that code for a kappa or a lambda light chain, genes that code for a heavy chain, and a gene that codes for a alpha, mu, gamma, delta, or a epsilon constant region. And the light chain kappa locus genes are located on chromosome 2 and is composed of 40 different variable kappa gene segments, 5 different J kappa segments, and a single constant K gene. And early in the development of the bone marrow, genes RAG1 and RAG2, along with the VDJ recombinase enzyme, work together to cut and join these different segments. They select one segment from the V kappa region and the J kappa region and transcript, splice, and translate it into a kappa chain polypeptide to be joined with the heavy chains. And then if a productive rearrangement doesn't occur like the first time, then it will try at the other K locus or kappa locus, and if that doesn't work, then it'll try at the lambda locus on chromosome 22. The lambda chain synthesis is similar to the kappa chain synthesis, except it contains 30 different V lambda variable lambda segments and 4 J lambda segments and 4 possible constant lambda regions. Once the variable lambda and J lambda segments are rearranged, then they are ready to be joined with a heavy chain. The heavy chain locus is located on chromosome 14 and contains approximately 40 different variable heavy gene segments, 25 different D heavy gene segments, and 6 J heavy gene segments. They are also cut and rearranged by VDJ recombinase and genes RAG1 and RAG2, which give the antigen specificity of the H chain. The VDJ unit is then joined together with the closest constant region genes, which are C mu and C delta, which code for IgM and IgD respectively. And then later on in development, when an antigen combines with a B lymphocyte and it activates it, it can occur a class switch recombination, which rearranges the constant gene to change to the classes of IgE to IgG, IgA, or IgE if needed as an effector cell. And then one process called somatic hypermutation increases the diversity and affinity of antibodies. It is the result of point mutations in the variable chain regions, changing individual amino acids. Generally, antibodies with a low affinity are produced in the primary response to antigen, but when they are further developed, the somatic hypermutation causes an increase in affinity to antigens. And this is referred to as affinity maturation. So the MHC is also regulated by a group of tightly linked genes that are passed down as a unit called a haplotype. The class 1 region is composed of genes on chromosome 6, which are HLA-A, HLA-B, and HLA-C, which code for the MHC class 1 molecules. It also includes class 1B genes, HLA-E, HLA-F, and HLA-G, which their function is not well understood, but it could be used HLA-E and HLA-F to present antigens to NK cells, and HLA-G could be used as a potential mechanism for preventing the rejection of a fetus. And there's also genes MICA and MICB, which are used in the class 1 region, which are also in the class 1 region, which code for stress-induced proteins that interact with NK and gamma delta T cells. And then the class 2 region contains genes that code for the MHC class 2 molecules, including HLA-DP, HLA-DQ, and HLA-DR. And then it also includes the gene HLA-DM, which codes for the molecule involved in peptide exchange in the exogenous antigen processing class 2 pathway. And then there's also a class 3 region, which contains genes that code for the complement proteins C2, C4, and factor B. The MHC is considered polygenic because it is made up of multiple genes that help with peptide binding specificities. And then it is also polymorphic because it contains different alleles of a gene, which allow the MHC to extend the range of antigens to which the immune system can respond to. And MHC class 1 and 2 molecules are codominantly expressed, which means they are transcribed from both the mother and father chromosomes. And every cell with a nucleus in the body actually expresses up to 6 different HLA class 1 and 6 different HLA class 2 molecules. And the expression of the molecules is what's called coordinately regulated, which means that all molecules are expressed at the same time on a single cell. And the set of class 1 and class 2 genes expressed on an individual chromosome is referred to a haplotype, which is passed down from parents to offspring as a unit, which means that children get different haplotypes from the parents, which creates a diversity of HLA.